what I'm going to talk about today, I decided I was thinking about this last night, and I decided that, what my talk is going to be on is really just the genesis of what I think is a pretty interesting project and something we're still thinking a lot about. It's not going to be maybe as much data as I usually put in these talks, but really trying to think about the students about, at least in my lab, how we think about problems and how we think about sort of moving a problem along in sort of a way that, that makes some sense and sort of really sort of stopping. I think you'll see in this project, finding a stopping point and really redirecting the way you think about something. And so hopefully it'll be interesting to you. So, my lab, we work on a lot of different things. We work on, a number of different bacteria, but one of the ones that we work on is Pseudomonas aeruginosa, and we spent a fair amount of time on this. This is what I worked on as a graduate student. And what we're going to talk about today is Pseudomonas in the context of sort of two environments. It was really nice talk by Gabrielle actually before, this talk, and it actually brought forth a lot of issues that environmental microbiologists, I think, are much further along than us who think about infections and work on human infections. And so the two environments that I'm really going to be talking about today are cystic fibrosis infection. This is actually a sputum sample you're looking at here. This is just naturally expectorated from an individual's lung. And then wound infections. This is a foot here, and then there's an infection here. And when you want to study these two systems, when you want to study CF or you want to study wounds, we use models. Almost everyone uses models to study human infection, for obvious reasons. And some of these may just be basic in vitro models. This is just Pseudomonas, beautiful green color, this bluish green color, Pseudomonas growing just in LB in the lab. This is a sort of, the mushroom biofilm, right? The way we used to think about biofilms, I think. And then there are other models, multicellular models. C. elegans is very popular, Drosophila, of course, mice. And then there's some host cells, models where you culture human cells often, and then you look at the interactions. We use these models, but there's really a question that we really don't know. And I think that is, how good are these models and how well do they actually mimic what's happening in a human? And, in my opinion, I think a lot of pathogenic microbiology has really focused on benchmarking all their data in mice, but not really benchmarking it in humans. And I think you heard Gabrielle say this a second ago that, you have to go out in nature as a microbiologist is studying, nature. You go out into nature to study microbiology. And we haven't really done that. It's almost embarrassingly little knowledge we have on actual human infections and the physiology of the bacteria. So today I'm going to talk about human infections in two ways. Primarily thinking about functions, and this word has been used a lot. Gabrielle used this a fair amount. When I say the word function, I just mean things like this. This is just Pseudomonas swimming. So swimming is a function. You might think another function might be, eating glucose or transporting an amino acid. Another function might be secreting a product. So when I say function today, those are the things I'm referring to. We'll also think biogeography, and the environmental microbiologists think a lot about this. This is very little known in humans about biogeography. But these are the two things we're really thinking about. And our goal here is to really think about these two things, these two ideas in the context of humans, and then asking the question, how well are our models recapitulating the functions of the bacteria in humans and the biogeography? And these are the papers that we'll mainly talk about. Almost all the work for me is from one postdoc. So the question is, how do we choose a model? So when you guys go out, we think about intuition, right? A mouse, it must be better than LB, right? For studying humans. I don't know that that's always the case, but that's what we think. Ease and flexibility. So, a lot of times maybe you need to do 5,000 assays or you need temporal data, and mice might not be the best place to do that. So you have to think of a model that you can use. Expense, whether it's dollars, euros, or pounds, right? Things, we all have to think about expenses. Reproducibility, this is probably the best reason why I think we use models, is so we can do experiments in my lab and you can recapitulate those and reproduce those well in your lab. some of you, for your student, the students out there, is just because you go in the lab and your boss tells you to do it, right? And your boss is telling you to do it because they've been doing it for the last 30 years. This is not necessarily a bad reason, again, because of reproducibility. And so that we can have expectations when new people come in the lab of the type of data they should. But then we get to this question is, those aren't necessarily the best reasons maybe to do models. I mean, they're good reasons, they're things we have to think about. But I think, when we think about models, we need to think about how do they actually, where are they good and where are they bad for recapitulating human infections? And like I said, what we want to do is we actually want to go out and benchmark our models in humans. We don't want to benchmark in mice. We want to say, this is what's happening in humans, and then use our models and say, how close are our models to what's happening in a human? So what you have to do, right, is you have to go out and you have to get human samples, and then you have to measure something. And so there's a lot of things that you can measure, but you want to measure something in these human samples, and then you want to go to your model and measure it, and then ask, how close are they, right? That's the basic idea. So what can you measure? Well, some people have talked about growth rate. It's actually been done in humans, but if you want to do this in a really broad sense, I think you need, more than a single measurement. And then you start thinking about omics, so proteomics. This is what I would really love to use, because I think proteins are, and for the most part, the functional units of a bacterium. And it would be really nice. Right now, it's difficult to do this in human samples. There's a lot of reasons why. Mainly is getting the bugs out, and so it's been very difficult. Metabolomics is another approach I don't think is very valuable. We can talk about that later. And the one that we actually settled on was transcriptomics. So actually going into these human samples that have Pseudomonas, extracting RNA, and then looking at the gene expression. So what I'm going to tell you today is a lot of work. It started about 5 years ago. And the reason it started 5 years ago is because taking debridement from this wound, where you basically cut out the flesh here and put it in an RNA stabilizing agent and give it to the lab, or this expectorate that you actually put into an RNA stabilizing agent. It took us about 4 years to actually get where we could reliably do bacterial transcriptomics. We have a very good protocol now, but it took a long time and it was a lot of money to do it. And it's a real difficult question, because you end up with a lot of human cells and a lot of human RNA. It's actually quite difficult to even lyse the bacteria, especially in the wounds. And if you look at our success rate currently, if you give us an expectorated CF sputum sample from a human that was directly put into an RNA stabilizing agent, we're pretty good. About 90% of those samples, if they have Pseudomonas, we can look at the transcriptome. It's much less for wounds. These are really, really tough experiments and really costly to do because of the sequencing depth. But what we're actually doing now is when we do the transcriptomics, we're using them as a readout for function. Now, there are certainly RNAs that have biological function, but obviously most of them encode protein, and we really want to think about the proteins. And so what we've actually do is we actually assume that an mRNA tells us something about the amount of protein that's there. And these are a couple of plots from papers we've done a couple of years ago, and lots of people have looked at the correlation between protein and mRNA, and it's okay. It's pretty good. There are certainly proteins that are disproportionately produced by, compared to mRNA, where mRNA is not predictive, and there's some that are. But that's a caveat to kind of think about as we go through this. So I want to start out by just telling you about this one study. This was the very first study we did. And these were the two folks that actually did this. Justine Dees was a graduate student in the lab. And then Dan Cornforth, who a lot of you may know, he was at Oxford and then he was at Edinburgh and then came to my lab as a postdoc, and he's now a research scientist. You can pretty much tell one of these people was doing RNA-seq and the other one was doing computational stuff. It's pretty easy to see who's who. And we started this study was with 15 human samples. We had 7 sputum samples, 6 chronic wounds. These were mainly diabetic foot ulcers that we had debridement from, and 2 burn wounds. All of these had Pseudomonas, and we did transcriptomes for these. This was a massive effort because this was the time where we were learning how to do transcriptomes. So this actually took quite a bit of work to actually get it done. The other things I'm going to talk about are some in vitro samples. This study actually used 87 in vitro samples from a lot of different labs, multiple strains of Pseudomonas, a lot of PA01 and PA14, the lab strains, they consisted of about half. And these transcriptomes were Pseudomonas grown in vitro in a test tube with antimicrobials or maybe with other bugs and maybe different oxygen. I'm also going to tell you about some mouse models. We actually have a lot more human transcriptomes for Pseudomonas than mouse transcriptomes now. We're going to get back to these in a second. Something to think about when you actually think about this study, and we actually submitted this as a grant and got a lot of pushback and ended up getting funded, but there's a lot of pushback. And the reason is because when we were doing this study, one of the things we were actually doing was we were taking human infection samples from different sites. And most people thought the Pseudomonas, the gene expression would be so different, right? In these different people with different sites. These folks were getting different antibiotics and different therapeutic regimens, and they had different co-infecting communities. And people were very doubtful that we were going to get anything useful from this. I was convinced we were, and I think we did. The other thing that you need to think about is RNA-seq. There's a lot of drawbacks to RNA-seq, and one of them not only is the fact that RNA doesn't always tell you about protein, about how much protein is there, but these are single time points, right? I mean, we just, one day, patient, individual comes in, we get a sample. And then the other thing that we're getting that you have to remember is that it's an average of all the gene expression that's actually occurring in the microbial population at the time. Now, we just got a grant in the lab to actually do gene expression of small populations and single cells from humans, but we're just now starting that work. So when we started, right, I mean, one of the big questions was just, no one had done massive human transcriptomics of infections. There'd been a couple that had published. There were currently none in Pseudomonas, I think, when this came out. And we just want to do a basic comparison of what's the gene expression like in the human and what's it like in vitro. And you can go back to this paper. It was published in 2018 in PNAS, and you can, there's an Excel spreadsheet that has 5,500 and, and 70 rows, and you can go through and see how it looked for your genes. But I had a much different question, because remember, the reason for doing this is thinking about, can we use this data in some way to tell us about our models? And so what we initially did, and I think it was a pretty good idea to start with anyway, it was definitely a starting point, is we used something called a support vector machine. So a machine learning, framework. And it's very simple. And it's basically allows you to actually ask the question, can you train a computer to distinguish between a human sample and an in vitro sample? How do you actually do this? You give the computer the answers and then let it determine the rules. So the answers here are the 80 in vitro samples, transcriptomes. You give it a transcriptome and you say this is in vitro. And you give it another, say this is in vitro. And you do this for all the in vitros. Then you go to the 15 human transcriptomes and you say this is a human transcriptome. The rules are gene expression, right? Asking the question, are genes expressed in certain ways that allow me, that are diagnostic for a transcriptome of whether it came from a human or not? It's kind of like your email filter in some ways, if your email filter works. Where an email comes in and you say spam, not spam, spam, not spam. The idea is that you learn what is spam and what's not spam. So then the question just became, when you run this, do you find anything? So can you come up with a set of genes that can reliably tell you if a transcriptome is human or not? So you can. With 30 genes, you're seeing the top 19 here. We'll go through them in just a second. But with 30 genes with over 98% accuracy, we can actually tell a human transcriptome from an in vitro. So you say, well, how do you know about the accuracy, right? Well, you leave some of the transcriptomes out. You don't give them to the computer and tell them it's in vitro or not. You let the computer come up with the rules, and then you feed it the transcriptome that might be in vitro, but you don't tell the computer, and you say, tell me if this is in vitro or not. And then you do that essentially with every transcriptome, and then you come up with an accuracy score. And it was about 98% with these 30 genes. So a couple of things that you want to look at. What this rank means here, this is the gene that is most discriminating between a human and an in vitro transcriptome. Okay, and then here are the other 19. This is, if you were to just compare in vitro to human, what would be the gene expression for this? So this gene is upregulated in the human transcriptomes about fourfold, right? Well, what can you kind of see from these genes? Well, the first thing, make sure that I'm staying on time. The first thing that you can actually see is that there's a lot of blank spaces in gene names. And what this actually means is that we don't actually know what most of these genes do. You've seen a couple of these genes. You saw one earlier, FUS here, it's involved in heme transport. What I was most worried about when we first got these genes is one of the things that really discriminates the in vitro from the human, right, is that all of these humans are getting antibiotics. Whereas in the in vitro conditions, almost all of those transcriptomes, most of them, Pseudomonas was not seeing an antibiotic. So you might be thinking, well, maybe these are just genes that are all regulated by antibiotics, and that's why you can tell the difference. And I was a little worried about this, and the good thing is we have a lot of information on Pseudomonas responses to antibiotics. And what you actually see is that only one of these genes, and it was only 2 in this top 30, were differentially regulated by antibiotics. So this made us feel a little bit better, right? That maybe this is something valuable that we're actually getting. And so you go, okay, so the computer can do this. And so how is this getting at your question? Right? I remember what our question was how good are models? Can we use this information? Well, so what we did is the next step was, well, we had all these mouse transcriptomes. And so then what we did is we actually fed the computer the in vitro and human transcriptomes, let it come up with these 30 genes. And then we fed it a mouse transcriptome and said, is this a human or an in vitro transcriptome? And it's going to put it in one of those categories. We did this for 3 mouse models. One of the mouse models was actually a burn wound model here. This is an acute model of burn infection for Pseudomonas. The mice actually die in this infection. This is a pneumonia model, which is also an acute model. The animals die. And this is a surgical chronic wound model where you basically make a surgical wound on the back of a mouse. This one resolves, so the mice don't die. What you're actually looking at here is fraction called as human. It's not exactly what this is, but it's the easiest way to explain it to you. But what you should realize is if this number is 1, that means it's classified all the time as human. And if it's 0, it's always classified as in vitro. Okay, so that's what you look at. What we see very quickly is that our acute mouse are very much more like, the transcriptomes are very much more like an in vitro culture than a human. Whereas in the mouse chronic model, it was much more human than it was in vitro. And you say, well, of course it is, right? Because you took all of your human samples from chronic infections, and that's true, right? And so this kind of makes a little bit of sense. But it does tell you that, you know, the mouse burn doesn't really do a good job of mimicking a human infection that is chronic. Okay, so let's say that you had a model, and we've had people do this, and they send them to us, and we do this, and we tell them, you know, oh, your fraction is human is 0.23. And so they're like, okay, you know, thanks. But what do you do with that, right? It's not really that valuable, right? It's interesting, but not particularly valuable. Well, you can dig in a little bit into the genes and start asking, why are these, for example, why is this mouse burn doing so poorly and the chronic doing better? And the way you do this is looking at the genes. Here are just a few genes, the top set of genes here that we're looking at. These are part of the 30 that allow you to tell in vitro from human. Here's their fold change in humans. Okay? Here's their fold change if you compare their expression in the mouse versus in vitro. So I want to bring your attention to these first set of genes. Well, here's the reason why the acute model, the burn mouse, is not doing well. And it's because none of these genes are expressed similarly to humans. Whereas in the chronic, they are. Okay? You can also see that there are sometimes that actually both models fail, right? These genes are actually downregulated. This makes some sense too, right? Because this is a gene inducible by antibiotics. We didn't have antibiotics in our models. But you can start to dig in, right? And it sort of gives you some nice insights into why your models, where your models are failing. And we've done this to actually infer some functions, but there's a problem. And the big drawback to this approach is that we're only looking at 30 genes. It's not really that useful, right? Pseudomonas has over 5,500 genes in the lab isolates. And so the other problem with this approach is it forces a transcriptome in vitro or human. So let's say that you have a choice and you can be in vitro or human, and there's this line, right, that crosses in vitro or human. And you might imagine just barely on one side of this line, you're going to be in vitro. Or maybe you just happen to just be barely on the other side, you're classified as human, even though those samples may be very close together. This is a drawback to this initial approach. I think this first paper was kind of cool, but it really left a lot to be desired, I think. And what the big goal was to go in and actually do something more comprehensive. And so what we wanted to do now is actually come up with a more robust genome-wide framework for doing this. So we want to do this with all the genes and actually be able to go in and provide really precise information to anyone about their model, about how well it actually mimics what's in a human. To do this, the first thing we realized is we needed more human samples. And because of that, all of this work now is focused only on sputum from cystic fibrosis. And the reason for this, right, is very simple. It's that the CF models work better. I mean, we can get a lot more, I mean, the CF human samples, we can get transcriptomes from easier, so we started here. We knew we needed our analysis, probably 17 or so really good transcriptomes to get this to work. Now, the other thing that we decided to do for this is we actually wanted to focus on 5 models, and all of these models will be using the lab isolate PA01. The reason we did this is because there's a lot of information on PA01, and lots of people use it as a model. So for me, it actually makes the most sense to start. So the CF models that we use were just some in vitro lab media, LB and a defined media that a lot of people use when they're doing biochemistry. We also used a synthetic cystic fibrosis medium. This is something my lab has been working on for about 10 or 12 years, and it's actually a medium that is developed in the lab. It's completely defined, which I think is probably its biggest advantage, is it's completely defined, and it actually includes a lot of amino acids and sugars and that are very similar. And the way we developed the media, I guess I should tell you, is we took a bunch of CF sputum samples and just did mass spec and biochemistry and figured out what's in it and then made it in the lab. The viscosity of this media is very similar to real sputum, and we've measured that. It also contains, you know, a lot of extracellular, a lot of DNA, and it contains mucins. And we use this model a lot. We also, there's an in vitro CF airway model. So this is a model, these are immortalized airway epithelial cells that are delta F508, so in CFTR, which you heard about earlier. And you grow these at the air-liquid interface and they differentiate. And you basically take the Pseudomonas and you put them up here on top, right, and let them interact. And the last one was just an acute lung model that's used in Pseudomonas. This is not the chronic model, this is an acute model. So we actually went out and we needed, so again, the first thing we needed was some more transcriptomes, right, from humans. So we went out and got expectorated sputum from clinically stable people. We took 7 from Copenhagen with Thomas Bjornskjold. He and I have an ongoing long-time collaboration. We did 13 from the US. Just wanted to have a couple of clinics, so we weren't clinic specific. And we sequenced these incredibly deeply. So in every one of these, we had over 4,500 Pseudomonas genes that had reads. So these are very deep and very comprehensive. And this is just a PCA plot, a principal component analysis. And you can see, I don't think this is that informative, but I showed it to you just because here are in vitro mouse samples on this principal component. You can see they separate, and here are the Denmark and US samples. So they cluster fairly well. All right. So now that we had our benchmark human data, this is our transcriptomes, these are our little gold, this is our gold, right? That we're gonna actually compare every model back to. Then we had to think about, well, how do you compare them? What's the best way to do this? And what's the most useful way? And I'll be honest with you, I'm not sure we've come up with it yet. I think this way I'm gonna tell you is actually interesting and useful. And it's easy to understand, which I really like it because I think it'll be used if it's easy. But it's something we call an accuracy score. I'm going to tell you how we calculate it. So the very first thing we actually do is we actually go in and we go to our human samples, right? So we have these 12 CF benchmarked Pseudomonas transcriptomes. And we take every gene that we have reads for that we can, that we can look at, and we look at the mean expression and standard deviation for those genes. When we do this, we can do this reliably for 5,564 genes. And what I mean by reliably is that these genes were getting reads from them in at least 16 of the 20 transcriptomes. This is so that's our benchmark. So we know what the average expression is, and we know what the standard deviation is. The next step we actually do is we go to our model, to the 5 models, we grow PA01 in those models, and we do transcriptomes. And then we calculate the mean expression of all the genes in the model. So you have the mean in the humans and the means in the models. And then what we do is we calculate what we've now turned an accuracy score for each gene. It's a z-score. And all we're really asking is this: how many standard deviations is the mean in the model away from in the human, right? So you have a gene in the humans, and it has an average, and we know what its standard deviation is. And then we have the mean in the model, and we just ask how many standard deviations away are you? And we call, the one we've been using is AS2, which I think makes the most sense, which is if you are a gene and in the model expressed within 2 standard deviations of how you're expressed in the human, we consider you to be accurate. If you're outside of that, you're inaccurate. We can argue about whether it should be 2 or 3. 2, I think, is pretty good. So we went through our 5 models and actually asked what the accuracy scores are for all these. So again, we used 5,564 genes. And here's what you actually see. So the most accurate model was our synthetic sputum that we made in the lab. Of the 5,564 genes, there were 783 that were inaccurate, that did not fall within 2 standard deviations. The CF airway epithelial cell model, right, where we grow CF cells and put Pseudomonas on top, was next. And then you see this is just growing Pseudomonas shaking in a test tube with in a defined media. This is the mouse, and then this is plankton. So this is kind of what you see. Maybe a little surprising, I think, that the mouse was as low as it was, but we'll get back to that in a second. So I just wanted to show you this a little bit of a different way. This is the same data, but just plotted out. Here, right, what you're looking at is standard deviations. This is 2 standard deviations. This is our AS2 measurement. And here is LB, the purple line. The green is MOPS succinate. In the epithelial cell model, the red, which is very difficult to see, is the mouse. And really the AS2 is where this is crossing this 2 lines, right? This is 86% for SCFM. Well, the reason I'm showing you this is not necessarily so you could see these curves, but what I do want you to see is this. And this is an important control. So what we actually did here is we actually removed pairs of the human samples. And then we calculated the mean in the other 18. And then we treated those human samples as if they were a model, right? And then asked how accurate are they? If you do that, their accuracy is around 97, 98% on average, which is this black line. So what this tells us and makes us feel good about is that a human sample as a model is much closer to another human sample. And you say, well, of course it is. Well, I think a lot of people weren't thinking that it was because they were thinking these human samples, right, could be so different between the humans. But I think this data is really tells us that, gives us really strong information that there is variance in human samples, but they're still very much alike. So knowing just the overall accuracy is interesting, but I think digging in is even more interesting. And you can start asking where things fail. So one of the things we can actually look at here is these are just tryptophan categories. So basically there's a set of genes that are important for amino acid biosynthesis, right? And we can look at, and so what's nice about this framework is we can actually look at the accuracy of subgroups of genes. And that's what you're seeing here. And so I'm not going to go through this, but here's our AS2. You see that, and what you see is that some some sets of genes do really well, like this purple line. Some do pretty poorly, like this yellow line. This is in our synthetic sputum. But if you actually look at the airway epithelial cells, the purple line does really badly compared to here, but the yellow line does better. So I'm showing you this because what you can begin to do is you begin to see that the models are failing and succeeding in different ways. The last thing I want to show you is another way of showing the data. I really like this way of showing, and I think it's very helpful. Basically what this is, here's an accuracy score color chart, where blue would be 100% accurate. This is all 5,564 genes. So this is SCFM, remember it's about 86%, so it's kind of right in here. But then what you do is you take these genes, you start breaking them out into functional categories like metabolism. And then you break metabolism up into further categories, and you get more and more granularity. And ultimately on the outside of this is just a ring of all 5,564 genes. But again, it allows us to really go in and find places, for example, here, polyamine biosynthesis, where SCFM does really poorly. And you can do the same thing with your epithelial cells. One of the last things I want to show you, I think I'm doing okay. Sorry. Yeah. One of the last things I want to show you is, well, here are the individual models again. And then these are the genes that are inaccurate. So here's SCFM2. Remember, it had 783 genes. Here is LB. It had 1,087, which is very close to the mouse. Well, one of the questions we started thinking, well, if we think about all these models together, are these models, do they capture most of the genes? Are there just a set of genes that no matter how we grow Pseudomonas, whether it be in a mouse or in a CFM or an airway epithelial, that we're just not capturing in this? And so it's pretty simple to do this. So what we began doing is saying, well, we're going to pair all these models together and see which two are the most accurate. And the two that are actually the most accurate are really interesting. If you combine the SCFM2 and the mouse together, what you see is they're only missing 358 genes between them. So what does that tell you, right? You have these models. I mean, the mouse is not very good, right? But what you know about the mouse is, while it's not doing significantly worse than SCFM, it is capturing a whole set of genes and functions that are not being captured here, right? So that when you combine them, you really reduce this down. And you can end up doing this for all the combinations. And here's the number, here's what I want you to look at. These are the all five models together. These are 211 genes that we did not capture in any of the models. We actually referred to these as elusive genes in the paper. So genes that are always inaccurate, no matter how we've grown in these models for PA01. So what are these elusive genes when you go look at them? The things CF people know a lot: alginate, I4 pili, O antigen, and a lot more. So a lot of things that we know actually Pseudomonas acquires mutations in when it's actually in a human, right? Kind of makes sense that that's why we're missing some of these. So how can you capture these things, right? Well, obviously, just don't use PA01, use a different strain, right? Maybe a CF strain. Maybe add another bug in. This is what everyone has constantly sort of asked us to do. And I'll be honest, as we've started to do this, the models have gotten much worse. And I really I'm starting to believe, and we don't have all the data yet, but just to anecdotally tell you, I'm really starting to think that the way that we get microbes to interact inside the lab are just not reminiscent of what happens and how they interact in a human. And inoculation methods. We're trying all these things. So I just want to give you one more piece of data. So can we capture these? We wanted to use a CF lung adapted strain. Makes some sense, right? So we just took the LES strain. Everyone in CF in the UK knows about LES. And everyone's asking me, why did we pick LES? We picked LES because LES is not in the clinical samples that we used. So this is a strain that was not in any of the humans that we used. And I thought it was a cool way to test this. So this is a distinct strain, you know, from different people in the UK, obviously in the UK, and spread around a little bit, right? But none of our transcriptomes were from LES strains. So we put that in SCFM2, and when we did that, we gained 112 of the 211 elusive genes. We captured them. They were expressed like they're expressed in a human. And we increased the accuracy up to 90% now. So I think, you know, what you're seeing is we're kind of on this genesis of a framework for helping us choose a model, coming up with a rationale. And one of the things that we're actually, you know, sort of really pushing into is we've done this now in CF, and we have a good sense of how Pseudomonas expresses transcriptome and chronic infections. We're going into acute infections, and we're trying a lot of other models like Drosophila and C. elegans. These are actually a little bit more challenging than I thought they were, just getting enough cells for a really robust transcriptome. when we're doing other mouse infections. But the other thing that the way to think about this is really a framework for improving a model system. And what do I mean by that? Just like using a CF strain, we've actually been identifying genes and pathways that are not accurate. And the beauty of Pseudomonas is that they're literally Pseudomonas grown in the presence of all kinds of different molecules. And by using and integrating that data in sort of these network analysis, one can actually begin to modify models. And I think I just got a large grant to do this with Jim Goldberg at Emory and Jim Bomberg at Pittsburgh. And it's not a great time to be hiring, I guess, especially in the US. But you know, we have multiple positions available. I'm going to be hiring 4 or 5 people, postdocs, research scientists, not just for this project, but I need to, you know, a lot of people leaving the lab and graduating, so I need a few more people. But the idea is, can we actually modify these models? And we are spending a lot of time thinking about this, and just, I really think, and this is my guess, that we can get SCFM2 probably to a 93 or 94% accuracy pretty easily, I think, with just a few additions. So this is kind of it. I mean, this is my lab over here, the Bitmoji of the lab. I think that's me, I'm not sure. You know, and this is them, my collaborators, Thomas, don't tell him. Thomas Bjornschot, Kendra Rumbaugh were really instrumental in getting me samples for this, and Mark Gilmore has always been a friend of mine that's worked with me on some of this. Arlene and Joanne are at Emory, and they help us get these the samples. The last thing I want to say is just that Pseudomonas 2021 is coming up, and Joanna Goldberg and I are the organizers, and it's going to be hopefully in person and in Atlanta. And here are the dates, and the Twitter, we actually just made it this morning, but there'll be more information coming out. So that's what I have, so thanks.
That was a really amazing talk. I actually presented some of your work in our journal club in the lab.
Oh, great. Okay. Well, hopefully you liked it.
Really enjoyed it. Okay, so we got some question for you. So someone's asking, when you obtain results from transcriptomes, how did you decide what is significantly? And yeah, so is twofold difference is important, or what cutoff you usually use?
Yeah, so, you know, for the machine learning, I mean, it uses, you know, that's a sort of distinct pathway. If you're talking about the basic DE-seq2 pathways, which I think a lot of people use, yeah, we use the statistics associated with that, and we try to be very conservative. But as a microbiologist, I never really trust twofold very much. I can tell you that we have followed up on a lot of the genes that you actually saw. The 30 genes, and there are, I can just show them to you. There are some that have been very fruitful, and we've learned a lot about. So this gene here is 4.6-fold increased. This gene is actually turns out to be what we think is in only a very specific human-induced gene, that is the switch to chronic infections in Pseudomonas, and that was about 4.6-fold. But it's really hard to really think about, you know, biological significance of twofold. Sometimes it can be important and sometimes it's not. And I just think you have to go in. I can tell you we verified a lot of these with RT-PCR, but yeah.
Okay, actually, just to follow that question, and something I wanted to ask, because in your transcriptal data, you normalized a lot of data sets from different background sets, but you also highlighted in biofilm, there's a spatial, you know, location of them. They probably genes expressed differently at different sites, different time. And do you think by doing that, you going to lose some of the fine-tuning genes involved in regulation? Or
yeah, for sure. I think that you're definitely going to lose that, and I think that's why I wrote this grant, you know, to go in and use smaller groups and do transcriptomes on single cells in smaller groups. You know, I think, you know, when I talk to ecologists, you know, I think there's value in looking at community scale expression, right? If we're looking at, you know, the functional outputs of communities. I can also tell you that we've done a lot of work with Thomas and others and submitted a paper that actually there aren't big biofilms in the CF lung. In fact, they're actually quite small. They're little groups. And in fact, a lot of the cells are planktonic in the CF lung. And so this idea that you have these very large biofilms in CF, sputum, is it's just, I mean, we really just don't see them very often. And so, but I think your point's well taken. You might imagine that it's not even a biofilm question. Maybe just two planktonic cells are very distinct, right, in gene expression. And so definitely something we have to think about. But this was hard enough to start with, I think. So I think we're going to go over the next hard stuff later. Yeah.
Very impressive work. Okay, another question. Do you have some non-linear methods like neutral network, or that's, I believe, it's for bioinformatics analysis and machine learning algorithm?
So are we using sort of network analyses to try to understand this? I mean, I think that is, I do think that it's a good way to do this. I mean, because I think, you know, we're going after things. And so where we're actually using those and where we're going after them, and this is really early days, but one of the things that we've done is we've actually gone in with chemostats in the lab and grown the bug under Pseudomonas under a lot of different growth rates and done transcriptomes. And then we're using these sort of, you know, there's some basic sort of regression approaches and other approaches that we're using to actually ask, can you use combinations of genes to actually determine what the growth rate is in an environment where you can't measure growth rate? And so those are certainly things we're going after. Those are good ideas, though.